Just leave this up so that people can join as they get on here, and then maybe in a few minutes we'll just start with who we have.
Professor Yost, this isn't for a grade, right? It's just review.
Right.
Okay. My cell phone's on, but my computer's dead. I have to charge it, so I won't be able to play, but I'll be on though.
Okay.
Okay.
I'll post the link for it later too, so if you wanted to play it like how we do the regular ones, you can.
Okay, it's fine.
Since over there, you there?
No, I think that's the only one that's open.
That's the only one. So
So there's no link for this Kahoot. You're gonna have to go to like the Kahoot app or website, whichever one it is, and you can type in this the pin number, or you can do the QR code with the link there.
Oh. 8234405.
Yep. So yeah, if you guys are just joining, or if you've never done like a live Kahoot before, I know a lot of people haven't. I don't think a lot of people use it at OSU OKC, but you will go to either the Kahoot app on your phone, or you will go to Kahoot's regular old website, and it'll ask you for the code, the PIN number, and this is the PIN number, the 8234405. You will enter that as the PIN, and it'll let you actually join in. As you can see on the screen, we have people joining in on this. So you can play actively, and you don't have to use your real name if you don't want to on this. It's kind of totally up to you what you want to do with that. So this isn't for a grade, it's just for review. And the good thing about this is, like, as we're playing, these are timed whenever we play them together like this. So you'll have like 20 seconds or whatever to answer a question. But I get a chance to explain to you why the answer is the answer, especially if more than one person is missing it, you know. So that's what I kind of like doing about with these reviews. It kind of allows us to stay on track with that and not spend too much time on some of the stuff. So it's about half of unit 1 and half of unit 2. So we'll go through unit 1 first, and then we'll get into the unit 2 stuff. And then if you guys want, I'll be posting the link to this. It'll be posted just like our regular ones that are graded, except of course it'll just be for review purposes. after we're done, so that if you want to, take your time with each question and go through it on your own, you're welcome to. I kind of like doing it with the group and having the timed version in this situation. In my other classes, or my traditional classes, whenever we have exams, I usually have 5 sections of about 25 students each. So it ends up being quite a lot of students. So we'll get a lot of people joining in, probably, for people that join in on reviews. Not everybody does, of course. So it'll end up being, I don't know, like 50 people that are getting on the Kahoot to do the reviews. And we'll usually do like the group competitions. And so then that's a lot more fun, and it has repetition of the information. The questions will appear multiple times for you, especially if you're missing them. They'll come up more frequently. And that's a lot more fun, but we just don't have enough people to do it, unfortunately. So. All right, I'm gonna go ahead and get everything started. Just FYI, when we have reviews like this, because the lab review is gonna be like this too. So if you happen to join in late, you can, the pin will stay at the bottom of the Kahoot, so you can come and join in at any point in time if you didn't join in the beginning. So just FYI, if that happens later. Or if you get kicked off, you can rejoin it that way. You might have to use a new name if you do get kicked off, which is why I don't do Kahoots for actual credit. They just are participation only. But yeah, let's go ahead and get started. I guess I can't see my start button. Sorry, let me get all this stuff organized. Right. So units 1 and 2, so overall, this could be microbes and how they function. Of course, unit 2 is going to talk more about how they function. But we'll start out with unit 1, and these were the chapters that we covered. Intro to microbiology with the tools of microbiology, then chemistry, and then of course, bacteria, eukaryotes, and viruses. Those were the big 3 that we covered in this unit. So let's get into it. So microbiology, what is it? The study of All right, great job. I'm glad we remembered that one. It's not too difficult. Organisms that are too small to be seen with naked eye. We need microscopes, microbiology. Not too bad. All right, true or false? All disease-causing agents are made of cells. Is that true or false? Yeah, so what you guys got this one, so it's gonna be false. And of course I used agents on purpose, so I didn't say organisms, right? Because we do have viruses and prions. Those guys are not even technically organisms really, because they aren't made of cells. So good job with that. All right, what is the correct term for disease-causing microorganisms? The ones that actually do cause disease, because excuse me, not all microorganisms cause disease, right? All right, so pathogen is correct one for that. Good job. If you're sick with a communicable disease, what's the one thing you know for sure? Thanks. You can see the pin now. Sorry, this, I don't know if you guys could see that bar that I have. Probably you can't, but I could. It was in the way. All right. So if you have a communicable disease, the thing that you know for sure is that it was caused by a germ. What didn't come out of, flipping nowhere. If you're sick with a disease, you can't say for sure that it was a virus, right? Because it could have been bacteria. What else could it have been besides bacteria or a virus? It could have been a fungus. It could have been a protozoa. It could have been a helminth. It could have been a prion, right? So there's a lot of things that it could be. Let's not forget about those other guys, but it had to be something. And that's why we say it had to be caused by a germ, it had to be caused by something. And in case you don't know what it's referring to, it's that germ theory, right? That Pasteur came up with. All right, the correct way to refer to the cause of the plague is Yersinia pestis, and look at how it's written. What system dictates how to write this? Anybody coming in late, anybody that decides they want to join in instead of just watching, you can always use that pin at the bottom, just telling you. Go to kahoot.it and enter that pin number. All right, so that's binomial nomenclature. Just gotta kind of remember that one. Taxonomy is a way to sort things out, but that binomial nomenclature is that two name word. So yeah, in taxonomy, kingdom is larger or more general than domain. Is that true or false? All right, so this one is false. Domain is the biggie there. Remember, for the domains, we have eukarya, bacteria, and archaea. So those are the three domains. And then those domains are split up even farther. So that's going to be into the kingdom, phylum, class, order, family, genus, and species. And you are going to need to remember those in order. So whatever works for you, like King Philip. I don't know what that is. Some people have something that they remember it by. I don't know. I just remember the sound of the words. I've said it so many times, but anyways. All righty. Inoculate, incubate, inspect, it's supposed to say identify, by the way. What is the missing of the 5 eyes? Which one's missing? That's what I'm trying to say. It's supposed to be 5 of them. All right, so this one's a pretty easy one. It's isolate, because we know we had to isolate things in the lab. We've learned about that term already. All right, what type of media allows only certain kinds of microbes to grow and will inhibit the growth of other kinds? It's a term, specifically that situation. Cool, so that's going to be selective. And don't get confused between selective and differential. It doesn't seem like you guys necessarily would. Most of you guys get that selective is inhibiting the growth. As long as you remember that differential is going to be what tells you the difference based on color, but it doesn't necessarily affect what can and can't grow, right? But can you have both in media? Absolutely. You can have media that inhibits growth, but also has a color change. Just remember that the color change isn't necessarily the thing that's causing the inhibiting, right? So the different aspects of those media. All right, true or false, the ocular lens of the microscope generates the real image. Which one, where's the real image being generated? And what image is the ocular lens generating? These should all be questions going through your mind when you see this. So the ocular lens is the lens that you're actually looking through with your eyes, ocular eyes. So that image that's coming through there is the virtual image. So why is that the virtual image? We have the slide that's sitting on the microscope. The light is shining through that. The first thing that light hits after going through your sample is going to be the objective lens. The image generated from that objective lens, therefore, is the real image. It's not been altered by anything other than magnify it. So that's a real image that we're creating there. Then we go through some mirrors and another lens at the ocular end, right? Remember, there's another 10x just at the ocular end of things. So that, because it alters that image one more time, is going to be the virtual image. So the real image is at the objective lens. The virtual image is at the ocular. If the objective lens is 40x and the ocular lens is 10x, what is the total magnification? All right, easy. So this is going to be multiplied together. It's 400x. It's pretty easy. As long as you guys, we know the multiplying thing, as long as you can actually look at a microscope and see, oh, it's on the 40x. And oh, when I look through it, that is at 10x. And it actually says that on the lenses, in case you guys didn't know it, and in case you forget. So you can figure that out, but ours are always 10x at the ocular lens, okay? So if you can see what objective lens it is on, and I asked you, what is the total magnification of this microscope, how it's set up here, then you guys should be able to look at the microscope and get that answer too. It's not, magic or rocket science. It's the same math. All right, what is the purpose of oil immersion? Why do we do it? All right, so reducing light scatter, you guys got this one, or reducing refraction, right? It's the same term. Refraction deals with light scatter. So true or false, a negative stain stains the background because it is repelled by the cell surface. Is that true or false? All right, so this is true. The negative stain, the stain itself has that negative charge to it, the outside of your cells. Remember when we talk about those phosphate groups on the outside, those phosphates are negatively charged. So they're going to repel one another. So negative stains stain the background. What type of chemical bond involves transferring an electron from one atom to the other? All right, so there's only 3 types of bonds that you have to actually know for chemistry portion, and that's going to be the ionic bond, the covalent bond, and the hydrogen bond. You can remember hydrogen bonds do not have exchange of electrons, okay? So they're going to be based on polarity attractions. So those are on their own. Ionic versus covalent. Covalent, we are actually sharing our electrons in the bond. And then ionic, we're creating ions, which those are going to be charged molecules, right? And the reason they're charged is because we have actual transfer of electrons from one atom to another. So giving charge to one, negative charge to one because they stole the electron, and positive charge to the other because they don't need that. So that's how that's going to work. True or false, carbon dioxide is an organic molecule. All right, so this one is false. The reason that this is false is just because something has a carbon molecule in it does not make it organic. It needs to have carbon that has a backbone of hydrogens. So those hydrogens are kind of supporting the rest of those bonds that are going on in that organic molecule. Is the most important aspect of that going to be the carbon? Yes, but if there isn't hydrogen supporting it, it's not technically going to be organic. But yeah, we even have things that are like carboxylic acid, which might be a H2CO3 or something, and that's not even technically an organic molecule because those H's are going to be given up as hydrogens and acid and all this. So it's more about, I'm not going to ask you about that one on the test, but it's more about the support of those hydrogens as well as the presence of the carbons. So not just the presence of the carbons. So olive oil is a great example of an everyday hydrophilic molecule, true or false? Okay, so hydrophilic, like Philadelphia, the city of brotherly love, where I went to graduate school, philic means love. So if it's hydro, water, philic, loving, so if water loving, it'll mix well, easily with water. Does oil mix well with water? No. So because we have that separation and that sort of, refusal to mix with one another, it's hydrophobic. So it's a good example of a hydrophobic molecule. Water fearing. All right, so which pH is the most acidic of these? All right, good job. So it's going to be 7. The lowest one is always going to be whichever one is the lowest. It's going to be the most acidic, even if you might think of it as being neutral. If it's lower than everything else, it's the most acidic. So. All right, which pH would have the highest hydrogen concentration? See if we can correlate this one. All right, excellent. So you guys are getting this. So the lower the pH, the higher the hydrogen concentration, right? The lower the pH, it's acid. In order for it to be acid, it has to have a lot of hydrogens in that solution. So you guys got it exactly. That's because of the way that equation is written as the negative log of the concentration. So that negative makes it inverse. All right, which level of protein structure features alpha helices and beta sheets? All right, that's going to be secondary. Most of you guys got that one. It's just a matter of going back. It's like these are basically terms we gotta memorize. All right, true or false? Bacteria and archaea are always single cell. All right, so this is actually true. Bacteria and archaea can kind of bind up with one another and create things like biofilms and whatever, but they are not true multicellular organisms. You can think of a multicellular organism like us. If I take, a biopsy of my liver and put those cells off to the side, are they gonna be their own organism? No, they have to be with the rest of the organism to survive. So even though we can have biofilms form with, and that's the closest we'll get to multicellular, but it's still even not, because they can still individually break off and still live as their own cells. So they are truly single cell organisms. All right, what cellular arrangement is featured in the image below? What most closely associated with this? All right, so the cellular arrangement, basically how those cells are organized with one another, grouped together, that's going to be strepto. Strepto is going to be chained together end to end. They're not always just straight chains. Sometimes we have branches of those chains, but as long as they're chained like that, that's strepto. All right, which flagellar arrangement is shown in the image? So we're looking at this guy here. And I'll just point out that we've got one at each end is how we're supposed to be looking at this. All right, so amphitrachus. So one at each end, amphi, both. You guys got it. All right, true or false? Gram-positive bacteria have a thick outer membrane. All right, this one is false. Gotta be paying attention to this wording. If you know that gram-positive have that thick peptidoglycan wall, and then the gram-negatives have the thin peptidoglycan with an outer membrane that contains endotoxin, right? Then those are the basics of gram-positive versus gram-negative. Gram-positive with their thick outer cell wall do not have an outer membrane. They only have that inner membrane, which all bacteria cells have, called the cytoplasmic membrane. All cells have that. All right. Next, all bacteria are capable of making endospores if nutrients are scarce. Is that true or false? All bacteria. All right, so this is false. Most of you guys are getting this. Which ones were the ones that only that were capable of making the endospores? There's only two groups, and that is Bacillus and Clostridium. If its name doesn't start with Bacillus or Clostridium, then no endospores. They're the only ones. All right, which group would Neisseria meningitidis, the gram stains can be pictured, fall in? So I would call this, just FYI, this is pink. If you saw the purple and what the gram-positives looked like, which we didn't have, we had some of them that were purple. You guys definitely saw the difference between the purple and the pink. But yeah, it'll be easy to tell in the test. It'll be very obvious when you see it. But so this was gram-negative because it was a pink or red background. So gram-negatives are going to fall into the gracilocutes, because remember, gram-positives, positive had the thick wall, so that's the firmicutes that makes them firm. Then I usually go to the Mendoza-cutes because Joey Mendoza was cute. That was a long time ago. So archaic. That's how I remember it, guys, every time. And that leaves us with gracilocutes and tenericutes. Gracilocutes starts with G, so does gram-negative. And then the tenericutes are what's left over with the ones that do not have a cell wall. That's the mycoplasmas. It's really hot in here. I'm going to have to take this sweater off. All right, true or false? Some eukaryotes can use cilia for locomotion or motility. So the eukaryotes here. That, so, yes, so they can absolutely. Remember, bacteria can only use flagella, but the eukaryotes are going to be able to do flagella, they can do cilia, they can do pseudopodia, which are like fake beads, like we see with amoebas, right? Right. What is the function of mitochondria? All right, so location of energy production. And if you didn't learn this from the eukaryote chapter, chapter 5, then you should have picked up on this from chapter 10, where we talked about it a little bit more. All right, which step of mitosis is pictured here? Okay, so PMAT, that's the order of mitosis. Remember, mitosis is for like making just direct copies of your somatic or body cells. So this is going to be something that eukaryotes do. Eukaryotes, like bacteria and archaea, do not do this. Okay, they divide by a totally different mechanism called binary fission. Mitosis is just for eukaryotes. So PMAT, prophase, that's how we're starting out. That's just consolidating all of your DNA into chromatin, making it nice tight bundles, and duplicate, making sure that the duplicated DNA is where it's supposed to be, and forming the spindles and all that stuff. Then MAT, M, is metaphase, middle. Just think of it that way. Lining up all those chromosomes in the middle, and then A, anaphase, apart, tearing those apart, and then telophase, we're going to make totally new cells. So you think that way, pinching them off and putting membranes around them and all that. Okay, so what is the term for the overall structure of an entire fungal mass? There's a term for it. Alrighty, so we know these are some terms, probably the 4 terms that I would say to be aware of for the exam. I know that septae is one of the terms that it talks about in like the textbook, but I'm not going to ask you about it because I feel like it is sort of a vague term as far as what we talked about with the fungi. But these terms, these 4 terms that were our choices here, I would know these terms from one another. Mycelium refers to that overall structure, the whole, how that whole fungal structure is sitting on your plate or on your bread or whatever. The hyphae is like when you look up close and you see that they branch off one another. Like if you zoom in real close to mold and you see that there's all these branching off of the hairs, those little hairs, as they branch off, each of those hairs is called hyphae. So that's kind of at a smaller level. Fungi can reproduce asexually and sexually. We don't need to worry too much about the sexual reproduction of these guys, but I will ask you about the asexual structures. And these guys will form either conidiophores or sporangiophores. Conidiophores are the open ones like we saw with aspergillus and penicillium, right? That looks like a little paintbrush or a dandelion, like the white ones that blow off into the wind that you blow and sends the seeds everywhere. They can look like those, like we saw with aspergillus and penicillium, but they're open. They don't have a membrane over them or anything. Sporangioforms have a covering that whole structure. So if you were to look at it and it had a cover, sporangiophore. If it didn't have a cover, conidiophore. Okay, so that's the only difference you need to know about those guys. Hyphae, the little hair branches, mycelium, the whole structure. Hey doggies, I hear you. Okay, what is the term for the free-living, metabolically active form of protozoa? All right, excellent. So this is trophozoite. Remember, for bacteria, we have the vegetative cells and the endospores, right? So those are their two forms. But if you have a protozoa, they're going to have the cysts, which is the dormant, hardy, surviving a lot of rough conditions. And then the trophozoite, which is the free living. And right, I've told you guys before, troph means eat. So if it's eating, then it is not dormant. It has to be the trophozoite is the active form. Okay. If you're dormant, you're literally dormant. That's how it works. So true or false, helminths are animals. Okay, so this one's actually true. It's just putting it into perspective for you guys. We've got a multicellular eukaryote, and it's not a fungi, it's not a plant, it's not a protozoa. It's actually going to fall into animalia. So yeah. It's got digestive tracts and all sorts of fun parts. Great. You can choose more than one of these. So select the types of genomes that viruses have been found to have. Yeah, so this is going to be all of these. It's just not meant to be really a trick question. And good thing is that on the exam, when you take it on the computers, just like the last time, there's not going to be multiple choice quite like that. It'll be one answer, will be the right answer. So you won't have to worry about missing one of the choices or something. So this is going to be viruses can have RNA genomes or DNA genomes. Those can be single-stranded or they could be double-stranded. And even when they're single-stranded, they could be positive sense or negative sense, which didn't really make sense until we learned about DNA in chapter 6. Remember, if you can read something as like a ribosome and spit out a protein, then you're positive sense. If you have to be made a copy first in order to read it, then you were negative sense. So that's what those mean. Yeah, there's a lot of different ways that viruses, it just has to be a nucleic acid. Any kind of nucleic acid. All right, true or false, all viruses have capsids. All right, so this one is true. The bare minimum for a virus is going to be the nucleic acid genome and the capsid protein that is like keeping it safe, basically wound around it, or icosahedron around it. So that's the bare minimum for a virus. Some viruses can have envelopes. So don't confuse the envelopes with the capsids. All of them have to have the capsids. Some of them can have envelopes, that lipid membrane that it steals from the host, right? Right. Where does the viral envelope, which I was talking about, come from? So I did just mention this. Let's see if you guys pick the right one according to that. All right, so the virus is going to steal it from the host cell, right? So when it buds out, and it like literally is like a little circle, and then here's the membrane, and it just pushes through, and then it pinches off that membrane around it to coat it. So it's just stealing your membrane. So how does it get its spikes, which we just saw in the image previously, the spikes that let it get in and out of the next cell, like get into the next cell, it's like their key to get in. So how does it get that on the outside? Well, it dots the outside of that host cell membrane with those spikes first, and then pushes through to coat itself. So naked viruses are just the viruses that do not have envelopes. So the ones that completely lack an envelope, that are just nucleic acid covered with those capsid proteins, those are naked viruses. If they require an envelope, they're not naked, right? They're coated. So those are the difference. Like picornaviruses, like parvovirus and poliovirus, which of course, if we were talking about parvo for dogs, very, very serious GI illness, could be very, very deadly for dogs. And then if we're talking about poliovirus, which has been almost eradicated from the planet at this point, but was extremely serious for humans, right? You had to be in the iron lung and all of this. And even people had permanent paralysis even after they recovered. So, and so of course, what it was the term for having the paralysis afterwards. That's sequelae, of course. I know I brought it up previously, but just to remind you. Anyways, those are naked viruses. They don't have envelopes on them. So they're super, super tiny. So we had to develop a way to filter out of water because poliovirus was so small, it was getting through our water filtration process. And so being a fecal oral transmission route for poliovirus, and it's only found in humans, not in any other species on the planet. Then in order to remove that and have safe drinking water again, we had to find a way to filter out that tiny, tiny, tiny little virus, one of the tiniest viruses ever known for poliovirus. So yeah, they're naked. So the naked ones can be smaller. So that's the one kind of thing they have going for them. All right, so true or false? Sorry, I can't read the front of this. The lytic phase is the phase when bacteriophages actively multiply until they burst out of the host cell. Is that true or false? All right, so this one is true. We have the 4 temperate phases. So that's a type of phage. You can be lytic phase or you can be lysogenic phase. So if you're going through the lytic phase, then you're multiplying your viruses, you're building your new viruses, and they bust out of the virus itself. Bacteriophages always bust out of the cell. They never bud out. So they are never going to have a membrane on the outside. They're not going to be enveloped ever. So none of the bacterial viruses are going to have an envelope because of that. So remember that. May or may not be on the test. So anyways, so they burst out of the cell after they multiply, great. Then go on and infect new cells nearby. However, lysogeny is going to be whenever they are going to hide their DNA in the host cell instead. And so they incorporate it in there. Then when the cell divides, remember bacteria divide a lot. When those bacterial cells divide, they're making copies of that prophage, that inserted virus in the DNA, every time that they duplicate as well. But whenever the virus is ready to, happy conditions, happy to pop out, then it will go back into that lytic phase. Sometimes whenever it sticks in there, it'll give that bacteria new genes as well. I'm sure that's what the next question is. I don't remember, but that's going to be lysogenic conversion, right? So what is the term for viruses that can cause cancer? So that was the next question. Right, oncoviruses, pretty easy. If you know oncology is the study of, treating cancer, then you'll know that this one's cancer. So it's going to include things like HPV causes cervical cancer. And they think there's a lot more that we haven't discovered. So what kind of genome do prions have? All right, so this can be none. Prions do not have any kind of genome at all. They're just misfolded proteins. That's it. And how do they infect and spread? Well, the misfolded proteins get near the non-misfolded proteins and cause them to misfold. So that's all. All righty, so we're going to move on to Unit 2. A whole lot of new concepts, and these are getting bigger, right? So genetics and metabolism are the 2 biggest ones that we have introduced here in Unit 2, for sure. The other ones are important, of course, as well, but those are probably the newest ones for a lot of people. So if Jenny has brown hair, and she has one brown gene from mom, but she has a blonde gene from dad, but her hair's brown, so her brown hair color is what? All right, so this is the phenotype. That's going to be what's expressed. If I said this whole BS to you and I said, okay, so her having genes for brown and blonde, what is that? Then you would say that is the genotype, right? Which one term describes DNA replication the best of the choices here? DNA replication. Okay, so in chapter 6, we have a lot of concepts introduced that I was introduced to them in like quite a great deal more spread out than what you guys are having to learn in one chapter. But DNA, we start out with replication, which is just when you have to make a copy of the DNA. Remember, when you are replicating DNA, just going from the exact copy of DNA to exact copy of DNA, then we're getting ready for mitosis. You have to have the copy and the exact copy, and they're linked together in the middle, and then we spread them apart. Remember in anaphase? Well, you had to have a copy made. So replication is what we go through in order to do that, so that those new cells each have a copy of exactly the same thing. So that's the importance of DNA replication. Anytime that you fall and scrape your knee, or you bump your elbow and catch a little blood, and your body has to heal, it has to make new cells. Those cells have to have the exact same DNA as the cells around them, as the ones before them. So we have to make copies of them. And that's all DNA replication is, making the exact copy. So that's one thing. Then a whole other different thing is using those genes for what they're meant for. And that's going to be DNA to RNA to protein, those steps. And remember, your DNA is what is in your nucleus, it's your genes. It's not doing anything other than having your genes. And the only thing that it's going to do is, make a copy of itself when the cell needs to divide. Other than that, you can open it up occasionally and make an RNA copy of whatever gene you're interested in. Now, remember, my example for this is having a big compendium of every single gene of your whole life in a Smithsonian institution. That's your nucleus, is the Smithsonian Institution. Can you go in there and take out your book from the Smithsonian Institution? You better believe no, right? It's staying in there, in the nucleus, in the Smithsonian. But if you want one copy of one gene to make, I don't know, blue eye protein, then you have to go in there and make just a copy of that one gene, and you'll just jot it down, right? So if it's written and typed out in your nice little genes, in your compendium, in your Smithsonian, when you're writing it out in your note to make a copy of just blue eye protein, well, you're just transcribing it, right? You're still writing like a scribe, you're transcribing it. That is that's your DNA to RNA step, transcribing. Okay, so you made RNA can leave. You can take that copy of just that blue eye protein out of the Smithsonian to go home to the endoplasmic reticulum that you live in. And then your ribosomes can come and read that copy out, right? So when it is reading that, it's taking what was words written down and turning it into protein, the product, the blue eye protein. Now, when you look at a blue eye protein and you look at the rules of how to make the blue eye protein, is that the same thing when you look at them? No. Is it in a completely different language now? Yes. So we've built from nucleic acid language that gave you instructions to the actual product of the protein that's built in amino acids. That's translating it into a different language. So that's the steps involved there. DNA replication, making copies of DNA. Okay, that's one thing. DNA to RNA, making a usable copy, a working copy. That's going to be transcription. And then going from that working copy, that messenger RNA, to make the protein using ribosomes, that's going to be translation, which I'm sure we're going to get questions about here in a moment. So I just wanted to clear that up, how those are all completely different things. And those are how I remember which one is which. All right, true or false, building blocks of DNA and RNA are known as nucleotides and consist of a phosphate, a pentose, and a nitrogenous base. Is this true or false? Oh, it's so hot. I should have put a fan in this room when I was redoing it. Okay. So we're about 50-50 on this. So it is true. So phosphate, we know we've got a phosphate on these, right? I've already told you, well, we've learned about how that DNA backbone is that sugar phosphate backbone repeating. And then popped off the side of those sugars, we've got a nitrogenous base, and that makes it, which nitrogenous base makes it A, T, C, or G for DNA, or A, U, C, or G for RNA. So those are the nitrogenous base and the phosphate, but what about that pentose? So we can say DNA and RNA are exactly the same if we list them out this way: phosphate, pentose, and nitrogenous base, because a pentose is just saying 5-carbon sugar. That's all that means. Penta, 5-ose sugar. Just like glucose, fructose, sucrose, whatever, ose, right? So pentose. So deoxyribonucleic acid has deoxyribose. That's the pentose there. And ribonucleic acid has ribose. That's the pentose there. So they're different pentoses, but they are just pentoses. Cool. All right, true or false? Exons are non-coding regions in eukaryotic DNA that must be excised to make a usable mRNA transcript. Let's look through the terms here, what we think might be important to know. These are the bulbs because I have LED bulbs. It's just hot in here. All right, so exons. We should know just by looking at that, if we're looking at this, exons and introns. That's just pop in your head immediately, all right? Which ones are which? Exons are the ones that are going to make the coding region, and the introns are the ones that we are going to be cutting out. So this is false because it should be introns are the non-coding regions that need to be excised, right? And we'll stitch all those exons together. The coding regions are at the exons. That's just a matter of remembering which one is which by memory. Sorry, I don't have a good way to memorize that. So what are codons used for? All right. What's a codon? What's it do? All right, so these are 3 nucleotide codes. So it'll be like AUG. So it's RNA, just to be clear, that's U, not a T. So AUG or CCG or whatever over 33333 over and over again. So those are codons. So there are 3 nucleotide codes in messenger RNA that tell which amino acid should be attached next when making a protein. So codons are on the messenger RNA. If I were to say triplets, the triplets are on the DNA. So it's the same code, the same 3, but on the DNA, they just call it triplets. On the messenger RNA, it's codons. And then the exact opposite of that messenger RNA that might say AUG, well, the opposite for it, that for A would be the U would be the A, the G would be the C. That's attached to a tRNA. And it'll come match up that anti-codon that I just described, which is the opposite, to that codon on the mRNA. And when those match up, that tRNA attaches the amino acid that it's attached to onto that protein as it builds during translation. That's how translation works. That all happens in the ribosome. So codon ACA codes for threonine. There's a mutation, it turns it into AGA. Now it codes for arginine instead of threonine. What type of mutation is this? All right, so this is missense. So we have, kind of 3 biggies down here that I had listed out, and then 1 rando, 1 fun sense, but silent mutations. If you had ACA and AGA, that went from threonine as your product, but still threonine after the change, that's a silent mutation. There would be an actual change in the genetic code, but not the product of the amino acid that's sent out. So that would be silent. This one, it turns out that ACA codes for threonine. We had 1 change from ACA into AGA. So that is now arginine instead. That's the wrong amino acid. And as a result of that can cause misfolding of the entire protein. It could be quite devastating. That's a missense. And then nonsense would be if you turn ACA into a stop codon instead. And stop codons, we call that nonsense because it doesn't code for anything useful. It just causes the whole thing to stop. And then you have frame shift. Frame shift, I know we had to learn about that, so I'm just going to go over it real quick, which is if you add or delete one nucleotide and it causes that whole reading frame to shift. If you kept all of the words in the book the same length, took out one letter in the second chapter, the rest of the whole book would be unreadable, right? Because you have to shift everything over. All the letters would be wrong, but the words would be the same length. And that's what a frame shift is going to do. Screws it all up. Okay, what types of DNA sequences do restriction endonucleases tend to cut at? Okay, so palindrome, so it's the same forward as it is backwards, like race car. So that's where it tends to recognize. What is the purpose of PCR? PCR, I'm just gonna let you know, polymerase chain reaction. I would know what that means. All right, so the whole point of PCR is to replicate a specific known DNA sequence. You have to, because as the image was kind of showing us, what we should know about PCR is the three steps of it being, denaturation, which is separating you have your two DNA strands, right? Like the ladder, denaturation just separates them, right? Then the next step is anneal. What are we annealing? Are we putting those strands back together? That's what anneal means, is to stick something together. So are we sticking those back together? Absolutely not. That's the point of this. So in great excess in the solution, we have primers. And those primers are going to bind at very specific sequences on DNA. So we are replicating only parts of the DNA. If you're doing the whole DNA sequence, that's going to take forever, even for like a regular PCR machine. So we aren't doing that. We're just doing specific genes that tell you that, yes, this is COVID. Yes, this is the plague. Yes, this is, the flu, whatever it is. And that you can tell each one from one another. It's only been very recently that we can do more than one in one run. Very recently that we can do more than one in one run. There's now one PCR that we can run in one sitting that'll test for the flu and COVID and the different strains of flu and COVID and RSV all in one go. And that's like revolutionary. So usually, typically, you're only going to test for one thing in one PCR run. So we're talking about a 3-hour run minimum, typically, let alone prepping the DNA for all that. And you have to have primers in that in order to replicate just the gene that you're looking for, which means you have to know what you're looking for. You can't just pop something into a tube and pop it in a PCR and then oh, this person has COVID. You had to put the primers for COVID in there. And you can't just mix a whole bunch of primers in there and say, oh, this turns out only the COVID one was used. They have COVID. So it's not typically how that's going to work. Anyways, so you have to have primers in there to target the gene that says that this is salmonella, this is E. coli, and whatever. And that's our known that we're working from. And those primers are key in identifying whatever it is that you're running this PCR on. PCR is not a magic fix-all. It's not going to tell you the answer to everything. I wish it did, but it's not how it goes, right? We have to have primers. We have to have primers. So that's what the anneal step is. We're sticking the primer onto that part of that gene so that the polymerase, remember polymerase is going to make a copy of the DNA, and like we saw in RNA replication, or DNA replication, excuse me. It's exactly what's happening in PCR. We're just DNA replication. Except we're adding on that primer for the DNA to polymerase to add on from the 5' to 3' direction, the nucleotides that are in the solution. And just doing that one little gene and doing it over and over and over again until we have enough to say definitively, yes, this is COVID, or no, it's not. We had no replication of the gene that we were targeting. And that's how PCR works. Okay, so it's DNA that we're going to be making copies of. And with each round, every single round, you're going from one double-stranded DNA to two double-stranded DNAs, because you're going one, split it apart, and then making a copy of it. Or I guess this is a better way to look at it. One, split it apart, and then making a copy of it. Then the next round, these two, split them apart, and then I'm making a copy of it. So in each round, you're doubling what was there when you started the round. So that's important to know too. We'll go over that again in the lab, because in the lab, I'm going to ask you after 4 rounds of PCR, how many copies of the DNA will you have? If you started out with 1 or whatever. So I won't ask you that on this lecture exam, but I will ask you that on the lab. Heads up. Okay. The second step of PCR is anneal, right? So what is being annealed? So let's see if you guys were getting it. All right, so this one, those primers are being annealed. That's what's being annealed, stuck on. Anneal means to stick it on. So our primers that are looking for the gene of interest are going to stick onto that single-stranded DNA. So we have the double strand. The first step, we denature it. In the second step, we anneal just the primers that are going to look for that gene we're interested in. So anneal it, stick it on. And then the next step, the third step, and there's only 3 steps really in PCR, it's going to be elongate, and that's just making that copy. So turning that single strand now into double strand again. Okay. It works on the same principles as DNA replication, just the gene of interest instead of the whole genome. Okay. In gel electrophoresis, what causes DNA fragments to be separated by size? What force is causing that to happen? Alrighty, so this one is electric current, so it is electrophoresis. The whole point of that is the electricity we're putting through it. We put your DNA in a gel. We push the electrical current through that gel while it's in a buffer. And the buffer is just helping that electricity move through there regularly and helping keep the gel moist and all of that. So the DNA is going to move through that gel because DNA is negatively charged, because the phosphates, right? So negatively charged phosphates, it's going to move to the positive end. Negative is attracted to positive, opposites attract. All right. You can choose more than one here. You may need to. BT corn produces proteins from bacteria toxic to certain insects, but they're not harmful to humans. So what does that make this corn? Alrighty, so if you are one of those people who thinks that they're avoiding genetically modified GMOs, I'm going to tell you, you absolutely are not. If you live in America and eat food here, you are not. Just end of story. If you've ever eaten at Chick-fil-A, you're eating GMOs. They recently switched to using antibiotics. That's a new thing for them. They were trying to avoid using chicken that had been treated with antibiotics, so they had to switch to that because of the market. So now they are using antibiotic, treated chickens to source for Chick-fil-A. But they are only sourcing from people who use antibiotics that are not used on humans so far. So that's something. But man, the market is inundated with products that I guarantee if you saw some of these things that you consume or buy or use or whatever it is, what they look like before they were genetically modified, you would not even recognize some of these things. What do you think bananas really look like before they were genetically modified? Because they don't look like what they look like now. Or how many seeds are in, some of the fruits that we eat now versus how big they should be. And there's stuff like, you know what I mean. And some of that's a problem. Of course, I'm not going to argue. Does that make it, I might be paying more or it might be paying less. but for crap, essentially, for some of this stuff. But then we have things like this that were meant for good, for farmers, which technically, that was the goal anyways. BT corn, probably 95% of all corn that you would consume here in America is going to be BT corn. BT stands for Bacillus thuringiensis, I believe. And the genes that are taken from that bacteria were inserted into corn to make them resistant to certain insects, and therefore not harmful to humans. Those genes shouldn't be harmful to us. I want to reiterate, I'm sure if you guys watch my lecture, they probably heard me say this a million times, but it's one of my biggest pet peeves, that when you eat something like BT corn that has a new gene put into it from bacteria, does that mean that you can get that gene in your body just by eating it? No. Okay, that's not how this works. What's the biggest issue with genetically modified organisms, with transgenic organisms, which are the same thing, by the way, which is what this question is also kind of getting at? The biggest issue is how they're going to impact our environment outside of these things. So we now have corn, that 95% of Americans, the corn that we eat is going to be BT corn. So what about those genes? Can they jump technically to other corn? They could. So then wild corn, because of the way that corn works, they have something called transposons, and they can jump genes. That's why some maize has like different colors of kernels. It's from they have genes that can jump that are for different colors. So this could do that. And then if you have all the corn in the whole world that's being resistant to certain insects, well, that's a problem, because that's part of the ecosystem and the way that the ecosystem works, right? So that's the issue with stuff like this, is the way that it can be affecting the ecosystem. Whether it's these genes that could be affecting corn around the world, or having the ability from plants to plants can have genes jump, even though it might not affect humans directly into your genetic code when you're eating it. The issue is that plants to plants, you could have a switch of some of these genes, and then now you're making non-corn plants like weeds resistant to insects, and then that is going to be a whole other problem, right? Or whatever, you get it. So that's the issue with GMOs and transgenic organisms, more than it is like, oh, you're eating something that has a gene inserted into it, and what do we know about what that gene does to that corn? That might be a problem. What do we know about the genetic modifications that could have occurred in chickens to make the chicken breasts larger and their thigh meat, more and stuff like that? Because chickens don't look like that normally. So how does that affect them? But then what proteins are they making that could affect us? So it's not the concept of the genes jumping into your body and causing you to have these genes in you. It's the effects of those genes having on that specific organism, and what byproducts they might make, and how it might affect the ecosystem and things like that. It's just stuff that we don't know. You won't know until you do, right? So that's the issue with GMOs and transgenic organisms. So spread the news if that is news to you. All right, true or false? Somatic gene therapy can be passed down to future generations. This is really asking you to know what somatic means. I said it earlier in the lecture. I wonder if anybody remembers. Alrighty. So somatic gene therapy is like your actual body cells. So if I give you gene therapy with an adenovirus or something that inserts new genes into your cells, and even it's like spreading it in throughout your cells and you're recovering and whatever, blah, 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 it generally isn't going to affect your germline cells or your gametes or your eggs or your sperm or something like that. Generally not. We can really only affect those cells because those are cells that are made very early on, especially for women, very early on in your growth in the womb. So we can really only target that if you're in the womb or before, like if we were doing IVF or something like that. So then we're talking about eugenics there potentially. Is that a slippery slope or what? I mean, we don't even need to get into that. If you've ever seen Gattaca, then you know that it is a slippery slope. But yeah. Okay. Next. What is the term for an organism that gets its carbon from inorganic sources and its energy from the sun? Energy from the sun, carbon from inorganic sources. So we got into chapter 9, we learned about this where organisms are sourcing their energy from. And there's two sides to it, where you're getting your carbon from and where you're getting your energy from. And then you put those terms together in order to make whatever you're referring to. So this is talking about inorganic, it's going to be autotroph, and the energy from the sun is going to be phototroph. If we got our carbon from like eating grass or eating other organisms, those are heterotrophs. That's where your carbon's coming from. And then our energy doesn't come from the sun, so it comes from the things that we eat also. So we are chemoheterotrophs. So those are the two I'd definitely be aware of. I don't really ask much about the others. Chemoheterotrophs and photoautotrophs, they're kind of opposites. All right, what is the term for a nutrient that must be provided in the diet because an organism can't make it themselves? This is one that I have on exams frequently. We even learned about it in Unit 1. All right, so the term for this is a growth factor, and just like the image was showing us, B12 is a growth factor for people. We have to take vitamin B12 supplements if we're not getting enough from our food, if your body can't absorb it from your food. But that saying that you have to absorb it from your food, you can't make it yourself in your own body. So it's a growth factor. You have to supply it somehow. Bacteria need that too, sometimes, other things, but. What type of solution could have a typical cell swell up and burst, just like this balloon? All righty, so the answer here is going to be hypotonic. So here's how we're going to think of it, okay? You take a cell, and again, typical cell, it's mostly water, but it's not entirely water, right? We know there's stuff in there. So let's say typically about 95% water or something like that. And it doesn't matter, but if you have a cell, no matter what its concentration is, in order for it to swell up and burst because you're swelling up, the only thing that can move across the membrane is water, osmosis. Nothing else, none of the salts or whatever, sugar, whatever it is that's around you can get in and out of the cell, only the water. That's how the membrane, which is selectively permeable, behaves. So water can cross it. We're talking about osmosis here. So osmosis dictates that because you have stuff inside of your cell and it's not 100% water. If you put that cell into 100% water, well, the cell's gonna say, I have stuff and I can't put the stuff to you to make this equal. So I can take in the water to dilute out my stuff. So it takes in water, it takes in water, it takes in water to dilute itself out, but it can't to a certain point, it'll just burst. So hypotonic. So on the outside, that tonicity, the stuff that is diluted in the water, would be like 100% pure water. It's hypotonic. There's no stuff in it, right? Low tonicity compared to the cell, okay? So think about what would happen to cells in hypotonic, what would happen to cells in hypertonic, and what isotonic means, which is equal inside and outside, right? So pathogens and microbes of our biome thrive at temperatures near or around room and body temp. So what does this make them? What's the term for that? All right, so this is going to be mesophiles. Meso means middle. So we're not extreme high or extreme low to us anyways, it's middle. So mesophiles. All right, what role do bacteria play in the nitrogen cycle? One of the more important things that we were learning about, at least in environmental microbiology, which I don't care much for, but I have to teach it, so. All right, so they're going to convert it to a form that plants can use. That's exactly what they're going to do. Take nitrogen that's in the soil and turn it into basically ammonia products that plants can actually use. And what do they use it for? Same thing that we use it for, right? What do we need nitrogen for in our bodies? We need it for our nitrogenous bases in our DNA and RNA, which we need a lot of in order to make our proteins, because proteins are made up of amino acids. And what is an amino group? NH2. So you need nitrogen for your DNA, for your RNA, and for your amino acids. So if you're making proteins, which is all your DNA even is for, and if you are being a cell at all, everything is controlled by your protein production, you're going to need a lot of nitrogen. So that's what we would need nitrogen for. Bacteria, without bacteria doing that for the plants, we would not be able to get our nitrogen from the plants and the things that we eat, which eat the plants a lot of times. So what are coliforms? We're getting there, we're getting there. Okay, so these are indicator bacteria. It doesn't mean that they are pathogens themselves. So you don't want to wait for the pathogens to show up, right? You're hoping that the pathogens aren't going to show up first. You're hoping that if you can test for just regular old E. coli or Enterococcus fecalis or things that don't normally cause disease, if you see these guys, which are present in fecal material, and they got through the filtration system, then there's a problem, because that means the bad guys like salmonella, like bad E. coli, those guys could get through, like cholera. Yeah. So you got these indicator bacteria that aren't necessarily bad. You can allow a certain amount to be present within drinking water versus what we consider like dishwater or gray water versus like pool water and versus like waste, treatment water, like from you treat your poop water, right? Being released back into the environment. There's different levels that you can have for each of these to have certain amount of coliforms. Of course, the goal is 0, but the world isn't perfect. So yeah, coliforms, just indicator bacteria. What is the term that refers to chemical reactions in the body involving breaking down molecules into smaller products? I'm sure you guys know. Yeah. Should give it away. All right, so this is catabolism, and that's how I remember it, because you have catabolism and you have anabolism. So of course, if you know about anabolic steroids, and anabolic steroids are what bodybuilders would take, to build muscle, and an anabolism, building, making new bonds, creating new things, right? Catabolism, cats to me are destructive, man. I don't have any cats, I have dogs and they're destructive enough, but cats, they've got claws and I'm allergic to them and everything. So they're just destructive to me. So catabolism, cats are going to destroy your breaking bonds here. So those are your two choices. Metabolism refers to all of the chemical reactions going on in the cell. So both catabolism and anabolism. And if you think about those, as we have learned about going into glycolysis, which I know we're going to get into, but taking glucose and breaking it into pyruvic acid, basically, right? 2 pyruvic acid, they're 3 carbon molecules from 1 glucose. It's a 6 carbon. And we can use pyruvic acid not just for the Krebs cycle and all of this and making ATP, but we can also turn that into amino acids, or we can break down amino acids and introduce them into the Krebs cycle as a step in order to end up making energy. And there's all these different steps that can go back and forth and in and out. That's amphibolism. So that breaking things down and then building things together and then going back and forth, that's amphibolism. Okay. And all of that is metabolism. All right. Cool. So enzymes help overcome activation energy of whatever the starter is into whatever product. So what's the term that we actually use for the starter? We talked about it in ELISA today. What do the enzymes act on? I did not bring my water in here at all. So enzymes act on substrates. So enzymes overcome activation energy, converting substrates into whatever their products are. So you could be breaking a substrate apart, So you have C and you're breaking it into A and B, or you could be taking A and B and putting them together and then turning it into C. Whatever your enzyme's doing, the thing that it's putting in its active site and acting on is the substrate, okay? That's our starter product. Cool, so it helps overcome the energy that is needed in order to go from A and B to C, or C to A and B, whatever you're doing. So what does ATP stand for? You don't know, this is our energy molecule. That's nice. What is it? And I won't put this question on the test. This is a little bit close. I won't make it this tricky, I promise. I'm glad that most of you guys got this right. You probably had to look it up, I'm not gonna lie, but I'm not judging because I'm not gonna make you choose between adenosine and adenine on the exam, okay? Just FYI. But it is adenosine triphosphate. That is because you have adenine, which is our nitrogenous base, right? We have osine says that there is a sugar group added onto it. So if it was just adenine triphosphate, there's no sugar in there. But we know there's nitrogenous base, there's a pentose, right? And it happens to be ribose, okay? And then triphosphate, 3 phosphate groups. That's where all the energy is, right? Because those phosphates pulling it apart from one another. But what does that sound an awful lot like? It should sound to you like a nucleotide with 3 phosphates instead of just 1, right? And that's exactly what it is. It's a nucleotide. So whenever I were to ask you what group of the macromolecules does ATP, our energy molecule, fall into, you should be able to say that this is a nucleic acid, because that's what adenosine triphosphate is. It's got that nitrogenous base, adenine, it's got the sugar, ribose, and it's got 3 phosphate groups. And those negatives pulling against one another, even though they're being held there by their covalent bonds, they don't like it. You can cut them apart, release a ton of energy to drive what the cell does. So in aerobic respiration, what are the 3 stages of energy production? So you can pick them out. Aerobic respiration. I already mentioned 2 of them. And yes, these all have to play in what we learned about in chapter 10. So R3, and you guys did really great on this one. I'm very proud of you for it. We're going to start with glycolysis, go into the Krebs cycle, and then the electron transport chain. That's aerobic respiration. So what are the other ones? Okay, so fermentation. When we do not have oxygen, we normally, if we were a facultative anaerobe, if we have oxygen, we're going to go through aerobic respiration. Okay. Oh, yeah, that's what they do. Facultative anaerobes, if they have oxygen, will go through aerobic respiration, just like you and I. However, if you suffocate them with a pillow, they're not going to die like you will. They will just go through fermentation because that's their means of anaerobic respiration. So what they do is, without oxygen, they just go through glycolysis over and over and over again. If you learned anything from glycolysis, you know that there is a little bit of ATP net that you're going to make, about 2 ATP net every time you go through glycolysis. Your glucose sugar goes through glycolysis and you make those 2 pyruvate molecules. Yeah. So they're going to make 2 ATP as a result of that. All right, cool. And then after that, their pyruvate that's left over, they had an electron that's released. It has to go back and make it ends up making acids and stuff, carbon dioxide and all the stuff that we see as products of what we think the goal is of fermentation. The reason that we get that stuff from fermentation is simply because they don't have oxygen to be their electron acceptor. They have to just use their pyruvate as their electron acceptor. So they ruin that guy and make all these products for us instead of going all the way through how we do in aerobic respiration to make ATP. Now they make 2 ATP from glycolysis. We do too from glycolysis, but then we also go through Krebs. We're going to get a couple ATP there also. Then we're going to go through the electron transport chain. All in all, going through glycolysis, Krebs, and electron transport chain for aerobic respiration, it's going to be 36 to 38 ATP. 36 to 38, not 2, 36 to 38, okay? So if you're bacteria, you'll get that 38. If you're us, you get 36 because we have to shuttle things across the mitochondrial membrane in order to do stuff in the mitochondria where our energy is made. So yeah, so that's how that is. So what are the other two? Gluconeogenesis, that is taking, if I eat a whole bunch of protein, if I'm on the keto diet, and I eat a whole bunch of protein, I'm not getting a lot of glucose to go into my aerobic respiration, then I can turn my amino acids and my proteins into energy and remake glucose to put it back in and go through energy making. Now you're going to lose a little bit of energy in order to make that glucose back up, but that's the whole point of it, right? So you can burn more calories, so you're getting, you know, as much as you're eating, you know, you're burning calories, basically what I'm trying to say. So to get the same energy. So that's why it's more effective that way for controlling weight and all of that. But anyways, that's gluconeogenesis. You're not starting out with glucose, but you can make it from whatever else you're eating. Gluconeogenesis, new genesis. Anyways, Calvin cycle, that is the dark reactions, the light independent reactions in photosynthesis. Okay. I'm sure we're going to talk about photosynthesis. I'm trying to teach you all about chapter 10 all in one sitting. So what is the final electron acceptor in aerobic respiration? Probably the most important thing to take home from all of this. It's called aerobic respiration. Aero, what does that say to you? This is where it comes into play. This is why you die if I suffocate you with a pillow. So you need oxygen. At the end of all of this, man, we're going to go glucose in glycolysis. We're going to glucose into pyruvate, 2 of them, right? Just 6 into 3. That's all that glycolysis is. All right. Then we're taking that pyruvate and we're turning it into acetyl-CoA, but that's a little middle step in there that we don't need to worry too much about. That's what's going to get shuttled into the Krebs cycle. All right. When in the Krebs cycle, what are we spitting out? What's the point of the Krebs cycle? If we say the point of glycolysis is to make pyruvate, the point of the Krebs cycle is to make electron carriers. So we're just shredding up that acetyl-CoA from that pyruvate. We're just shredding it up in the Krebs cycle and releasing out carbon dioxide as waste and stealing off electrons as we're going and sticking them onto our electron carriers, FADH2 and NADH. Those are electron carriers in aerobic respiration. So great, we've got electron carriers. What the F are they for? They're going to take those electrons and pop them into the electron transport chain. In the electron transport chain, which is our 3rd step in aerobic respiration, we're moving electrons across the membrane through cytochromes, these proteins. And as the proteins are reacting to all that, it shuttles across protons. Because you remember how I told you it was FADH2 and NADH? Right, they're carrying those electrons with those H's. So it turns out if you take the electrons free from those H's, what do they make? You have the electrons free. The H's are now H pluses, right? Protons. So as protons get shuttled that are left over out of the membrane, out of the membrane, out of the membrane, as those electrons are moving through the membrane. Negative, 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 positive, 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 creating a proton gradient outside of the membrane. Now, we've created that positive gradient on the outside, right? What we've learned about with osmosis and gradients and everything like this, diffusion, is that those positives want to equalize on both sides of the membrane. They want to come back in, right? So we should put them through a channel, and the only channel that they're going to be coming in through is called ATP synthase, and it does exactly what it sounds like. As those protons are coming back through there, it causes that protein to change, and then that allows us to put phosphates onto a DP, adenosine diphosphate, and turn it into adenosine triphosphate. This whole process is called oxidative phosphorylation. We're not spending energy to make this ATP. In fact, we're using just the negative charge of the electrons and that positive gradient that it's allowed us to create in order to drive that shape changing of that ATP synthase to make our ATP, our energy for the cells. So we've got all these protons coming back in, trying to equalize the gradient. What happens if it equalizes the gradient? It can't make more come through. We can't make more ATP, right? So how do we keep that going? So to get rid of those H pluses that we're building up, to get rid of those electrons after they move through the whole thing, we don't need them anymore. We take the electrons plus the H pluses, direct to H's again, right? And then we take our good little old friend oxygen, take half of that sucker and put 2 H's onto an O and make water. So if we don't have oxygen to clean up that mess that we're making from making our ATP with those H pluses, with those electrons, if you don't have oxygen to take care of that so it doesn't equalize on both sides of membranes, then you die because you can't make ATP. And it happens so fast in your whole body where all of your cells might be making ATP that if you get suffocated with a pillow, within a couple of minutes, you die because you don't have oxygen to help you take care of those electrons and those H's that have built up at the end. That is why you die. Think about that. I think that is the freaking coolest thing of all time, you guys. You learned about that. And now you understand it about how that works. Is it so simple as, well, you die because you don't breathe in oxygen? Well, yeah, but tell me why. Why? Why do you need the oxygen? What is it for? Now you know. Now you know why you need oxygen. And that's also how come we can say, whenever you're using that oxygen, it makes what as the byproduct? Water. It doesn't make carbon dioxide as the byproduct. That oxygen that you breathe in does not turn into carbon dioxide. The carbon dioxide that you breathe out, all of the carbon dioxide that you breathe out, all of that carbon dioxide that you breathe out comes from the Krebs cycle and that acetyl-CoA BS, turning into acetyl-CoA from the pyruvate. Pyruvate into acetyl-CoA and then going through Krebs cycle, that's where all of your carbon dioxide comes from that you exhale. That's how important this is for your body to function. I can't stress it enough. That's why metabolism chapter, I feel like, is probably one of the most important chapters that you're ever going to learn about, as far as physiology, which I don't think that, no offense to Dr. Shearer, I don't think he goes into enough detail on. So. All right, true or false? Fermentation generates just as much energy per glucose molecule on average as does aerobic respiration. What did I say about fermentation earlier? All right, so this is false because, yeah, fermentation, when you don't have that oxygen that I told you at the end, right? And you still got an electron that you're going to make in glycolysis, because everybody, all organisms have to go through glycolysis. All living cells have to go through glycolysis, all of them. So even if you're an anaerobe, if you never touch oxygen a day in your life, you still go through glycolysis. But our fermentative are ones that go through the fermentation stuff. If they have oxygen, they'll get their 38 ATP from this whole thing by going through aerobic respiration. Our fermenters, our facultative anaerobes, will go through aerobic respiration if there is oxygen. So don't ever forget that. If there is no oxygen for our facultative anaerobes, our fermenters, then they will ferment. So what do? They just go through glycolysis and they stop there. They never go through Krebs. They never go through electron transport chain because they don't have oxygen to deal with their electrons and their protons. So they just keep it there at glycolysis. Glycolysis only yields 2 ATP. If you're not going through all of aerobic respiration, you're not getting all of the ATP, all of the energy. So it's impossible for it to have just as much per glucose, right? Fermentation per glucose gets 2 ATP, whereas aerobic respiration per glucose gets 36 to 38 ATP. So not even close. Okay. Next, based on what we've learned, which organism would be a true pathogen? We're getting real close, I promise. All right, so the answer to this one is Yersinia pestis, because it always causes disease. Streptococcus pneumoniae is one I can understand if you aren't aware of. It does cause meningitis and pneumonia, but it also is a normal inhabitant of the body, just like staph can be, just like Candida albicans yeast, right? These things are normal to have on the body in small amounts in the places where they belong, just like E. coli. E. coli is normal in your gut, but it doesn't belong in your urinary tract, right? So Yersinia pestis always causes disease, so it is a true pathogen. Okay, virulence factor, what is it? This is one that comes up a lot, so especially in, well, all the coming chapters, I talk about this one a lot, so it's a good one to understand. All right, so this one is something that increases pathogenicity and infectiousness of an organism. So anything. So what if you have, we've mentioned this previously, if you have a flagellum, if that helps you make your patient more sick, then that is a virulence factor. If you have a capsule, if it helps you evade the immune system, then that is a virulence factor. If you make a toxin, obviously that's a virulence factor. But anything that helps with pathogenicity and infectiousness qualifies as a virulence factor. And whenever you guys are filling out your disease PowerPoints, I know you're probably going to get to them after the exams, so that's on you, whatever. But whenever you're filling that out, don't just say like one thing for a virulence factor. If there is like, you know, a whole lot going on with that organism, don't just say it has virulence factors or something. Be aware of what virulence factors are and show me that you know what they are with your organism. Another thing I want to warn you guys for your presentations is if you are telling me about how to treat that thing, don't say you did a blood test. Because I'm sure, as you well know, that this course teaches you what tests we run on the blood, and I expect you to tell me which tests are done to test your organism. Like if you were working on COVID, you might say there are PCR tests done on nasal swabs, as well as at-home tests that are based on antigens. So this will be to any disease-causing pathogen at all. So it's not just going to be, so virulence factors apply to viruses, yes, but also to bacteria, fungi, protozoa, helminths, and prions. So any pathogens. Okay. All right, which term refers to an agent which came from outside the cell or the body? All right, so exogenous, exo being outside. Good stuff. It could be more than one for this one. So which of the following are symptoms? Select all of the symptoms. All right, good job. So most of you guys getting this one, it's gonna be the headache and the fatigue. Remember that symptoms are gonna be things that are kind of subjective. The patient is telling you that they're in pain, for example, and how do you measure that? How do you prove that? So headaches, fatigue, you can't actually, you might kind of be able to tell somebody is tired or that, but you can't actually physically verify it with your fingers or measuring or something like that. So lymphadenopathy refers to swollen lymph nodes. You can actually feel if somebody has swollen lymph nodes, and a fever, of course, you can measure with a thermometer. So those are going to be signs, not symptoms. In which phase of infection are the very first signs and symptoms seen? There's 4 phases to an infection. They're all here. Where are we going to see the first signs and symptoms? Alrighty, so it's gonna be the prodrome. Good job, guys. Remember, it's gonna be incubation. Your body hasn't responded to it yet, and most of your symptoms are coming from your body's response to the organism, whatever it is. And it's probably not multiplied enough yet in order to cause problems. So that's the incubation period. Next is the prodrome. Your body's just starting to respond to it, probably a lot of innate immune response. And then we have the acute or infection phase. That's going to be like the height of the organism multiplying and causing disease. So it's the worst of it. So you're actually full on sick. And then convalescence is the decline of the symptoms as you're clearing the infection. All right, a fly flies out of the porta potty and lands on your food. It's not infected itself, but you might get sick. What is the fly? All right, so this is going to be a mechanical vector because it itself is not infected, which would make it a biological vector. But it is a living thing, so it's not a fomite. So fomite are non-living things like a cell phone. Like if I had COVID and I use my cell phone and then you're like, oh, can I use your phone to check blah, blah, blah? And I'm like, sure. You touch it, you touch your face later and you get COVID. This was a fomite. It itself is not infectious, but it can be a vector, right? It can transport disease. Mechanical vectors are whenever you have a living creature that is sort of acting like a fomite, okay? That is spreading the disease. Okay, who was involved in identifying the source of cholera infection in the 1800s London and containing it? What's this individual's name? Okay, so this is John Snow. Yeah, we just gotta know his name. Not that John Snow, obviously, but old time John Snow. All right, so this could be more than one. Which term refers to the occurrence of a disease in a given time frame? We're looking at the number of occurrences in a month, let's say. All right, so this actually has two answers. If we're looking at how many infections popped up in the last week or in the last month, specifically just in that time frame, then that is looking at incidence. That's a certain time period. Like we've had 15 infections in the last week in Oklahoma for COVID. I don't know how many we actually had, but that would be incidence. That is the exact same thing as morbidity. Okay, mortality is how many deaths we've had, and prevalence is talking about how many we've had up to now. So all of them up to now, not like in the last month or the last week. It's going to be all that time. I think that's one of the hardest part with the epidemiology part is knowing the difference between incidence and prevalence and remembering that incidence is the same thing as morbidity. So cholera outbreak in the 1800s is where people got sick from the water pump over several weeks. What is that an example of? Okay, so this is technically, good job, guys. This is technically going to be a common source epidemic because we are looking at cholera coming from that one pump, yes, but over a long period of time, maybe a few months or something. However, if we had a point source epidemic, if I came in with a new strain of COVID in class and I'm all like talking to you guys in lab and coughing on all you guys in lab, and I'm spreading my new strain to all of you guys, and all you guys go home and you end up getting sick, that's a point source epidemic. You saw me at one point in time, and all you got sick at that one time. It was not spread out over several weeks or something, right? So common source says it would be like over a period of time. All right, outbreak that extends exceeds the expected prevalence, but which does not cross continents, is known as. So we have an outbreak that exceeds the prevalence than what we expect. So we might see a couple cases of this normally, but now we're seeing a whole lot. And it didn't cross the continents. That's an epidemic, exactly. If it crosses the continent, it's pandemic. If we just had a couple cases, but we don't normally see it, but it's not going on for a while, then that would just be sporadic. An endemic is if you're used to seeing a couple cases and you're only seeing a couple cases, that's normal, endemic. All right, multi-select, this could be more than one. What does it mean for a screening or diagnostic test to be highly sensitive? I hate the difference between the sensitive and the other one. Oh, okay, so it's two of these, and the other two are for the other one. So this is going to be for sensitivity. Test trending towards identifying any possible positive. So if you have like if you're very sensitive to hearing a sound or something like that, you're very sensitive, you're gonna hear it pretty much the second. Like I hate hearing people eat or chew or whatever. I can't stand it. I will friggin' hear it from across 10 miles away or something, I swear. So that I'm very sensitive to it. Other people might not even notice it. So I will identify any specific case of that. And it's the exact same thing here. So yeah, it's also going to be gearing towards false negatives being less likely because there could be somebody chewing across the street that somebody else might not identify, but because I'm so sensitive, I will. So I'm going to hear out all of these people chewing with their mouths open, right? So it's that sort of thing. Just being literally like sensitive. If you're sensitive to something, then that's what this would be talking about. However, if you're being very specific, which is our other one, you are going to only recognize positive. So if I'm super, super sensitive, but not specific, I might hear random noises that I think is chewing and start getting mad about that, right? But if I am both sensitive and specific, then I'm not going to get mad at just random noises. I'm only going to get mad at the chewing noises, and I'm going to hear all of them. So I would be both at the same time, and you can have both, and that should be the goal with your test making. But there are two different traits that are distinct from one another. Sensitivity, you're going to catch all of the possible positives there could be. And then, yeah, so, yeah. I don't know what else to say about it other than that, but you're looking for both of these, but they are different traits from them. Does that make sense? Oh, so hot in here. All right. What is the term for how many people an infected individual is likely to infect during the course of their illness? You got sick? Sorry. No, sensitivity and specificity are not inversely related. Sorry, I know that you guys are probably trying to answer this other question, excuse me. But we had a question, are sensitivity and specificity inversely related? Absolutely not. You can have high of both of them, but they are just separate traits that you have to be aware of. And it's sort of presented that way, in the way that, like, I feel like the lecture sort of presents it, because they're so just like distinctly like on the same kind of taught at the same time, and just shown as like one is looking for the false, you don't have any false negatives, and this one has no false positives. Well, you want neither, right? You want no false negatives and no false positives. So you want to have high sensitivity and high specificity, but you but they are separate traits from one another, right? That's all that is trying to say. If that makes sense. I hope that answers the question. Sure, right, back to this one. What is the term for how many people an infected individual would infect? This is R naught. This is just one of those terms that we have to kind of be aware of. This is a memorization term, right? So R naught. All right, so that is it for our review. Okay, that is it for our review. Do you guys have any more questions for me before I let you go? I am going to post this quiz as like a normal Kahoot quiz for you guys if you want to use it. As soon as the video is done processing, it does take it a while, but as soon as it's done processing, I will post that as well. Yeah, I think that's it. I hope this was helpful.
Yes, thank you.
All right. Yep, I mean, I'm glad to help. I know it's a lot, especially in the summer, so, You guys-
Mr. Rose,
yes.
is there a difference between a mechanical carrier and a passive carrier?
Um. That's a good question. And we could think of a passive carrier as being like, sometimes we have mosquitoes that'll drink blood, right? That's infected with something, but they themselves still aren't getting infected, right? And we could think of that as being like a mechanical, but because it's like in their biological bodies, I would think that could be more like a passive carrier. But I wouldn't worry about just like worrying about the distinction for our course, right? So that's how I would think of it. Being a passive carrier, you're a mosquito, you're now carrying blood from somebody that's infected, but yesterday you weren't, right? But you weren't infected with it yourself. But it's not the same thing as a mechanical vector, like what you see with cockroaches or with flies or something, right? So yeah, there's a little bit of a difference there, but we don't need to worry about that for this course, really. I'm just wondering for you guys to know the difference between mechanical versus biological, if that helps for that part, but. You guys are really good about asking questions for actual understanding. Appreciate that.
Could you go over the difference between septicemia and bacteremia?
Yes, so if you have any bacteria at all present in your blood, it could just be like a passive transmission, right? You might not have a true infection in your blood with the bacteria, but you shouldn't have any bacteria in your blood, right? So that's bacteremia, but if there's an actual infection, if that bacteria is flourishing, truly growing, causing infection within the bloodstream, then that is septicemia. Does that make sense?
Textbook, I think it just says septicemia is the rapid reproduction of and a large presence of microorganisms. It doesn't
Yeah, it
really
could-
differentiate.
Yeah, so it could be any microorganism if it is actually truly infectiously reproducing within the bloodstream itself, then you could consider that septicemia of any kind. So you could have that way. Septicemia doesn't have to be bacteria. It's just we typically really only see it with bacteria, most commonly. So you don't see really septicemia occurring with like fungal infections or something. You've got a whole other problem going if you've got that going on. So I feel like, yeah. But you do see it quite frequently with bacteria, right? So that's why I focus typically on that. But I don't want people to get bacteremia confused with septicemia, simply because bacteremia is just, if you had just even passive amounts of bacteria in your bloodstream, from a contaminated blood transfusion or an injection from needle sharing or something like that.
So bacteremia suggests just a little bit of it in the blood. It doesn't have to be necessarily multiplying, whereas septicemia is large amounts and they are actively flourishing.
Exactly, yes. That's what I would focus on is basically the presence of bacteria versus the actual multiplication and dividing and growth of the bacteria. Good question.
And focal infections they begin as localized infections, but something happens to a pathogen and it spreads out from the initial contaminant site, correct?
Right. And there might not even necessarily be a sign that's going to spread, everywhere or even become septic or something like that. But yes, it would be spreading. Like if you have a sinus infection and then that leads to having, pneumonia or something like that. That could be, because it's not spreading all over, but it makes sense how it would have traveled to that area of the body. All right, anybody else have any other questions? Okay, I'm gonna go ahead and head out then and work on trying to get this stuff uploaded for you guys. If you do think of any of the questions, you can email me and I will try to get to them tonight. If I don't notice anybody's email and you email me tonight at like 2 a.m. or whatever, then I will try to get back to you right before the test, because you know, it's fair game to ask a question up until the moment we start the test, so just FYI, if it comes down to it. So, yeah. You guys don't have any more questions, then I'm gonna get out of here. Oh, I never showed the freaking podium. I'm so bad about this. Nobody ever reminds me to show the podium. Anyways, yeah, you guys are doing really good compared to my traditional classes. I mean, honestly, I'm not kidding about that. I'm not just trying to butter you up or something. My traditional classes suffer a little bit more than what you guys are doing. You guys are putting in the work and it shows, so. Keep it up. All right, I'm gonna kick you guys out and I will see you guys tomorrow. Best of luck studying, best of luck on the test.